Hello, and welcome to the Boffin Buzz, the podcast for anyone fascinated by farming's future. Well, hello, and welcome to the next episode of the Boffin Buzz. And I'm absolutely, we're talking science today, and I'm absolutely delighted to welcome Pete Iannetta to the Boffin Buzz today. Hello, Pete, how are you?
Hi, Tom. I'm doing fine, thanks. Yourself?
I'm very well indeed, thank you. The sun is almost shining. It's something we haven't really seen very much of lately. But never mind, we're not going to moan about the weather today, Pete, because we want to talk about science, and in particular, the work that you have been, that both of us actually have been doing on the NCS project, which we'll talk about in a little bit. But the first thing, I wanted to find out more about yourself, Pete, because now you work for the James Hutton Institute, is that right?
Yes, I'll be there 30 years this April.
30 years. Wow, yeah. Fantastic. Well, look, tell me, how did you get started in science? You know, what drew you into that particular field then?
Mainly because I love being outdoors originally, which is kind of ironic now. I spend most of my days on a computer, but I do. I did like fishing and whatnot. I was a hunting, shooting, fishing type, right. And went off to St Andrews University originally to do marine biology, but I ended up changing during the course of my time there to plant biology and ecology, mainly because of a chance meeting. I was in charge of a social group. We invited various scientists in to give talks, and this amazing lady turned up called Janet Sprent, who's a professor in legume science. Right. And she gave a talk during the term time, and I thought she was just bowled me over with the kind of work that she was doing. She was somebody right. who, even in the 70s, was writing letters to the press saying, "Why the hell aren't we eating British beans?" I think that is when I was brainwashed. I went to work for Janet over the summer. And cutting a long story short, eventually I ended up doing a PhD in biological nitrogen fixation by legumes, with her, and didn't really like the university environment. No offence to university boards that are out there, but I looked at the James Hutton Institute, which was the big house on the hill down the road from the university, and I liked the applied work that they do, and also right. the fact that it informed political decision making. So I veered, my interest veered more towards the more applied research as opposed to, something more academic.
I see. And so has your interest always been in proteins then, Pete?
I would say it has. That doesn't mean I've always been possible for me to research that. Because when I first joined the Institute, I was in the pathology department working right. on diseases of soft fruits, but also potatoes. And continued on in fruit quality for about 8 or 10 years. Throughout that time, though, I was always doing legumes in the background. It's always been a theme that even when I've not been specifically funded to do it, I've kept, a finger in that pie. And particularly with the economic crash 2008, with the environmental crisis, the importance of legumes for environmental and economic stability, has increased. So since about 2008, the economic crash, it's been obviously a sad event, but it's sharpened people's senses, shall we say, with respect to resource use efficiency.
Well, I must say, Pete, I mean, I don't want to get too drawn down this road, but I mean, as a researcher in legumes, you must have seen, the current, crisis, we might say at the moment, climate crisis coming from miles away. And you must have been sitting there thinking, legumes, for goodness sake, when are we going to start talking about legumes? And yet my impression is that they suffer from quite a bit of underfunding when it comes, and yes, particularly on the research side. What are your feelings as far as that's concerned?
Well, we exist in a global marketplace, and that's just the reality of our time. So as long as legumes can be imported cheaply, it will probably continue to be done. So the truth is, things like the health benefits of pulses and legumes can be gained through imported grains. However, you do forfeit the benefits that can be achieved in the environment. They
Yeah.
can only be achieved by growing them. Where we are, certainly I do feel at times, well, the feeling about it, it's an actual fact that many of the costs of food consumption are not borne out in the cost of the food itself.
Yeah.
And that's just a reality of our time. But I do believe that the import door for pulse grains should not be as opened as widely as it is.
Well, do you know
Uh,
what? That leads us very neatly into the NCS project, or Nitrogen
mhm,
Climate Smart, as it's known for short. I won't spell out the entire project title. Regular listeners of the Boffin Buzz podcast will be familiar with our Pulse project that we are doing. This is a £5.9 million DEFRA-funded project with 17 project partners led by PGRO. Specifically looking to raise the homegrown pulses, so both on arable side, but also the use of pulses on farm. So our aim, our bold and ambitious aim, is to increase to 20% inclusion of pulses, peas and beans, in the arable rotation, and it's currently about 5%, and also to raise by 50% the inclusion of homegrown pulses in livestock diets, to move away from imported soya, as you so rightly talked about just now, Pete. So those are the ambitious aims. One thing I always stress at this point is that we're not going to do that within the 4-year life span of this project. We don't expect suddenly that everyone's going to start growing pulses. But what we are doing is we're working with 200 farmers, and we're looking to carbon baseline them. We're going to talk a bit about that in a minute. And we're also recruiting, at the moment, we're recruiting our Pulse Pioneers, who are going to be doing on-farm trials and having a look at the productivity of beans on farm and trying to raise that. And we believe that knowledge has value, that those farmers, the knowledge that they have about growing pulses, if we can improve that knowledge, then it will help everyone raise their game on pulses. And through the 200 farms that we're baselining, we hope we'll be able to demonstrate that by the end of this project, just the benefits, the sort of benefits that we're talking about within this project. And the benefits are huge, aren't they, Pete? I mean, I think the cost benefit alone is we've worked out as something like, if we manage to achieve what we want to achieve, we can reduce input costs by about £1 billion a year, I think,
mhm,
isn't that right? And
mhm.
then there's the reduction in carbon emissions. Just remind me, how do you remember how much we're aiming to?
The exact number escapes me at the moment, but I can say that it's around about half of the emissions that UK agriculture is responsible for.
Yeah,
So half of that would be, if you like, a quarter of the emissions would be dealt with by stopping import to soybean, and
that's just incredible, isn't it?
the other quarter would be dealt with by other additions. It could be
Mm,
soil carbon sequestration, it would be fertilizer offset, and a whole lot of other things. We don't perhaps want to get too caught up in. I mean, carbon is absolutely a right thing to be focused on. Carbon footprints. We're in a climate emergency after all.
mm,
Um, but there are a number of other challenges as well, and I do feel that with pulses we can deal with a number of challenges simultaneously.
yep,
We could be dealing with, for example, crop diversification and biodiversity loss. We can be dealing, of course, with the carbon emissions, and we can also be dealing with nutritional provisions, improving the
yeah.
quality of nutritional provisions, not just for animals, farmed animals, but also for humans and for aquaculture, which is another big potential user of beans.
Great. Well, now one of the aspects that I know that James Hutton Institute is involved in is the Centre for Sustainable Cropping, and we're looking to maximise the benefits from that platform with the Pulse Pioneer trials that we're about to get underway. So, can you just give me a little bit of background about the centre first of all?
Sure, the CSC has been running for 13 years now, and it was conceived a long time ago um, on the basis of some farm experiments that had gone on where the fields were studied in a split field design, with one half of the field being managed conventionally and the other half being managed with new innovative methods. So we took that split field design and we have a 6-crop rotation, a faba bean based rotation. We also have legumunda stories in the form of clover to cereals. And because it has a split field design with conventional and innovative methods on the other half, it's strictly scientifically controlled. We have 300 different indicators that we look at because we're really interested in ecosystem function. And it's not, as I said, the innovations that we implement are integrated. That is, they're stacked innovations of various forms, and they're all designed to reduce pollution, maintain biodiversity, including non-crop biodiversity, the arable food web, the insects and bugs in the soil and whatnot. And we want to obviously maintain yields or even increase yields with minimum inputs. So we have for a long time been a leaf innovation centre, and so it's an integrated platform based on the leaf, if you like, design.
That's the linking environment and farming. Is that
That's
the new?
correct.
Yeah.
That's correct. So we haven't gone down any particular route. We haven't gone down conservation agriculture. We haven't gone down organic.
Right.
We wouldn't like to be trapped by particular routine practices. So we do all of those things as necessary to minimize input. So we will stack innovations as we see fit.
So sorry. So
Yeah.
just so I'm clear, so that the idea of the platform is you're kind of testing a rotation, but what you're doing is comparing what you might call a standard rotation, a typical sort of farm standard rotation,
Yeah.
with what you would regard as a more innovative approach, which is based on ICM.
Organic inputs, lowering fertilizer application, using integrated pest management, having buffers at the side of the field to limit runoff and soil loss, for example. We have magic margins that we've deployed that also helps biodiversity and runoff.
Right.
We're looking at different varieties as well. We use minimum tillage. The residues that are left over from the crops go back onto the field. We also have compost that we would put on from time to time as well. So there's to put it into practical context, we're able to maintain conventional level yields with 25% synthetic fertilizer use reduction.
Right.
So
I see. So hang on.
Mm-hmm.
So this is so what we're saying here, Pete, is that this system works. The system that you've got works. I mean, is it like one rotation, or are you trying lots of different methods? I mean, because I can imagine that it's horses for courses when it comes to trying new innovative elements within your rotation, and what's right for a farmer in the Lincolnshire Wolds isn't necessarily going to be right for someone, on the Norfolk Sands, for example. So, have you got lots of different rotations that you're
No,
testing?
just one standard rotation, quite typical for the arable northeast.
Right.
With the usual crops in, quite a large barley representation. That's 2/3 of the rotation. We've got our oilseed rape, we've got wheat, potatoes, and faba beans.
Right.
Yeah.
And then the rotation that you're comparing that with
is exactly the same rotation. So the only thing that's different is the management, and they're not using integrated pest management. They're not putting in high levels of organic matter. They're going with rates of fertilizer that might be recommended. Whereas we'd be below that. Instead we might be using other techniques. As I said, we mentioned IPM, but it could be biofortification. We had a slight issue with our wheat yield on the integrated side where we stacked the innovations, and we found that biofortified fertilizer deals with that issue quite inexpensively and quite well. So it's about developing other solutions to the problem that means maximizing our ecological functions, on the integrated side. And obviously, there's other results there as well. For example, we're finding the soils are becoming naturally disease suppressive. we have less leaching
Okay.
of nitrogen, obviously, into the waterways. So there's eutrophication, nitrogen pollution, phosphorus pollution that's avoided, and whatnot. So it's been interesting watching. Also, another curious thing we found recently with our because we can analyze full rotations, of course, is we found that yield stability has become much, much, much tighter on the integrated side.
Really?
So yeah, and to me, I think for farming that really is quite significant. So we're building yield resilience into it. So that platform is managed by a good colleague of mine called Kathy Hawes, and she gets input from our farm staff here at the institute with regards to its running and keeping it real. Yeah.
I see. So, how are we going to bring the benefits that you've seen within the CSC trials to our farmers into our Pulse Pioneer trials? Have you had any thoughts on this, Pete?
Well, one of the beauties of the CSC is that we monitor the system. I'd love to see more
Yeah.
farmers monitor the system, and it's not so we can police them. It's just so that they
Mm.
can evaluate, what it is that they're managing, the direction they're moving. Quite often, we're all too busy to look at what is happening in the system that we're managing. So whether it's something very simple like soil carbon levels or water
Mm.
pollution levels, it could even be other things like specialist insects and pollinators.
Yeah.
But I think being aware of your system gives you an informed, position to go from. So at the moment, the CSC is terrific. We've got all these very, very practical functional indicators that relate to system function, including the economic functions as well, I should add. It's
Yeah.
not just that we're studying the environment, how it stacks up economically has to be there too. Um, and we're aware that a lot of people are going daft with life cycle analysis, which is a very useful tool. It tells you how much impact you're having, and hopefully you would hope year on year your impact to be reducing. But as a scientist, um, there's a misfit there in that the assumption with a life cycle analysis is that because it's an impact assessment,
Mm.
is that your reduced impact means the function is being improved. Well, I would like to see evidence of that as a scientist,
Right.
because rather than just assuming. Well, for example, if an analogy I sometimes use is, well, Tom, I could kick you 10 times today, and you might not like that very much. Um, and you can't walk after I've kicked you 10 times, but tomorrow I'll only kick you 5 times. So that's better, isn't it? Even though perhaps you're still not able to walk. So, from my point of view, the point is how well are you functioning,
Okay.
and that I can't assume you can't assume that a reduced impact is going to lead to the ecological improvements you'd like to see. So,
Right. Okay. Well,
yeah,
before we descend into complete violence, no, I have to say it was a slightly loaded question I asked
yeah,
you to start with because, as you know, with the Pulse Pioneer trials, what we're asking our Pulse Pioneers to do is follow their crop through yen and all the detailed analysis of the crop that yen provides throughout the year and throughout the growth cycle. So that's and what we're asking the farmers to do is not only look at their pulse crop, but also within their pulse crop, we're asking them to have an area that's not grown to pulses so that they compare one with the other. And so they compare it within the season they're growing the pulse crop, but then they monitor it in years afterwards to see what effect it's had on the rotation further down the line. And
mm-hmm,
then within their pulse crop, we're also encouraging farmers to look at interesting things that they can do. Perhaps some of these interesting new biostimulants and things like that they can apply, and then also look at it in some detail. And I hope that the hope that we all have really, Pete, isn't it? That through looking at these crops in some detailed analysis, we can get a more granular understanding of what it is, how we can raise productivity levels, and it's that consistency of yield. I think that most farmers I talk to feel that they're not getting from their pulse crop at the moment. Which,
mm-hmm.
if that's what you're getting on the CSC program with some of your programs at the moment, then that's what we want to replicate on farm, really, isn't it?
Well, yeah, absolutely. It's a shame sometimes that I feel that legumes are blamed for not being good enough.
Yeah.
I hear quite often people talk about yield instability, in post crops,
Mm.
and actually there's been a number of studies now that would question that. I don't think the yield instability is any worse than any other crops, but I would say that farmers perhaps need more experience
Right.
and
Well,
more years working on legume crops. But also, I would say that legume crops are quite environmentally dependent. You know, they're not like the other crops. They don't perform well under high nitrogen, for
well,
example. But
let's
yeah,
dive into that a little bit
yeah,
more,
yeah, yeah.
Pete, because I want to exploit your knowledge, your expansive knowledge on legumes. And when we before we started this podcast, you told me about or you mentioned briefly their complexity, and you said how
Mm.
and so on. So tell me what you mean by this. Tell me about legumes. What
Well,
do we need
we've
to
recently
understand?
published a scientific article. Basically, it's entitled "More Than a Meat Substitute" or
Mm,
nitrogen fertilizer replacement. And the provisions of legumes are much more complex and actually phytochemically very diverse. You know, and when you grow them, whether you take the grain and consume them or use it as a whole crop forage for animals, or the residues are returned to the soil, they make a very organic provision. Now, when I say organic, I mean complex provisions, phytochemically diverse. Obviously, there's carbon there, obviously, there's nitrogen there, but there's a whole host of other chemicals that are
right.
going in. And I think that makes a difference to soil function. Also
Right.
can make a difference to gut function. An approach we're using with some of our research at the moment is to look for solutions that allow us what we call One Health options, and that is a way of cropping to give yields, including residues that go back to the soil that are both good for the environment as well as good for ourselves or our farmed animals. So taking a One Health approach is something I think I'll be doing a lot for the next 5 or 10 years.
Right. I
Yeah.
see. Well, so it's about, it's not just about growing the legume and the
Mm,
nitrogen benefit that we all know
mm.
about that you get from that. There's a whole other set of factors that they bring to the system, and not just the arable system, to the livestock gut as well. Is that what you're saying?
Yeah, yeah, absolutely, and including it could be the aquaculture gut as well.
Yeah.
That's they can be big users of faba beans and peas and whatnot. So absolutely, I think we're looking at multifunctional approaches. We've got quite a few things we have to deal with in our civilization. Obviously, the climate change is one, but we also have to deal with biodiversity loss. So crop
Yeah.
diversification is a big challenge
Mm.
going forward. Also, nutritional provision, the complexity of that. We know that diet diversity also leads to longevity and improvement to the quality of your life as well. And the same can be said for farm animals as well. Yeah,
So, how much do we understand about that? I mean, what I'm understanding from this, Pete, is that legumes provide a huge sort of very sort of complex array of the, whatever they are, factors, chemicals, and so on and so
mm,
forth, and natural uh, biofortification, whatever
hmm
pathways to
hmm
our system. And that's not just to our soils and to our arable systems, but also to our livestock, to
hmm
ourselves as well when we eat
hmm
them. How much do we actually understand about that? In terms of the specific benefits that they're providing. Are we just sort of touching the top of the iceberg here, or do we now sort of pretty much understand what's going on?
There's always we never understand it all. Nature's such a complex beast, but what I
Right.
would say is that it's well proven that uh, legumes grown properly have what's called a symbiotic microbiome around them.
Right.
So the microbiome is that collection of bacteria that live around the roots. Now, the
Mm.
very obvious ones we know about are the rhizobia. They're the rhizobia that actually they form an association with the legume. They
I'm
form the
glad you
legume
mentioned
root
the
nodules.
rhizobia, Pete, because that's one thing that I'm really keen to talk to you about because you've mentioned it in conversations in the
Yeah.
past to me, and
Well,
I've always
we can
said,
come back to it, but
rhizobia.
going back to those complex provisions that
Yeah.
legumes provide, you have this symbiotic microbiome that relates
Mm.
as much to the soil function as well as much as the rhizobia itself. So um, I think it's good to keep that in mind. I was touching on it earlier on the fact that legumes don't do well in a high nitrogen environment. Um,
Yeah.
from my experience, they do well actually in mixed systems. It's a worry to me that farmers have become increasingly specialised, and as they have become increasingly specialised to deal with certain markets, they have by default entertained an ecological inefficiency by not having the animals on their farm with the crops that they grow. Because the animals also make a complex organic provision to the system function as well. I feel that pulses are grown best in mixed systems.
Well,
Yeah.
actually, I tell you what. Let's just establish one particular thing right here and now, and that's the use of livestock manure with pulses. Because I know it's a controversial thing, so we are not allowed to apply livestock manure to a pulse crop. And I suppose that, on the surface, the arguments are pretty obvious. Well, it provides its own nitrogen, so why
Mm-hmm.
would you? But at the same time, it feeds the rhizobia, and as far as I understand, and I'm sure you'll correct me if I'm wrong, manure is good for pulses, but there's a but. Now, can you just talk me through this? Why is it that it's not a good idea to put manure onto a pulse crop, and does that mean that manure is not
I
good for
think you have to
pulses?
be very, very careful. Manures come in lots of forms.
And just a brief introduction for any Pulse pioneers who may be listening to this podcast. If you'd like to claim 2 Project Ambassador points, please get in touch with Sky and let her know the code phrase, which is life cycle analysis. That's life cycle analysis. Now back to the podcast.
Certainly from all the experiments I've done on nitrogen application to crops like faba bean is, yes, they can still nodulate, and yes, they can still fix nitrogen, but actually the performance is not good. If you want to optimize legume performance, you do it in a low nitrogen environment.
Right.
So although animals may be in those systems, as you say, they're not applying manure directly to those crops. You're quite right to highlight that. But nevertheless, animals in the system means there's provision somewhere in the rotation at some point for things to be added into that, complex organic additions to be made. I suspect one of the key additions that animals make is not so much in the form of nitrogen, but phosphorus.
Right.
And it might be that's having the effect. But there's bound to be a whole load of cascading effects because of the effect on the microbiome, just with the presence of these organic animal manures, which once they are in and mineralized, the system is a different system to one which would not have animals in it. Look, going back to the CSC, our system didn't get to where it is overnight. It took at least 6 years to evolve into its current stable state.
Right.
And it would be the same for any system. It doesn't adapt overnight generally.
I see.
But it is. I mean, to put it into some sort of context, the yield could be reduced by half by adding nitrogen to pulse
Really?
crops. Yeah, if you add
Right.
excessive amount, and the lower your soil nitrogen when you have a pulse crop, the better it will perform. And we see that as well in intercrops. If you put something that grabs nitrogen from around a legume, like a let's say a barley or a wheat, the legume will fix more. Its protein content will be higher. It's responding to the fact
Mm.
that the non-legume with it has depleted the soil of nitrogen.
Okay, well, let's dive
Yeah.
into that a little bit more. But just to conclude that conversation, so I think what we're saying is mixed farming systems, livestock, and specifically the manure that they provide to the soil is good for pulse crops, but not directly in front of them. I think we can agree.
Yes, yes,
So
yes.
that's great. That's
Yeah.
cleared up one thing. But let's dive into rhizobia because this is something that I'm really fascinated about, and I know that you've done a lot of work in this. So tell me a little bit more about that relationship that pulses have with the bacteria in the soil.
Well, it's complex. Yeah. So this association isn't quite as old as the association legumes have with mycorrhizal fungi, but it's a little younger than that. But still, very, very ancient association. Not every legume crop nodulates with the same rhizobia, so that's one thing to keep in mind. Although peas and beans do generally crop well with the same type of rhizobia.
Sorry, could you
If
just first of all explain, Pete, what you mean by nodulates with rhizobia? What's going on there?
very complex series of interactions. So the legume is secreting chemicals that the rhizobia will recognize. Chemotactically, the rhizobia are free living in the soil. It's a bit more complex even than that. Some of the rhizobia in the soil have an element to form a symbiosis, and some don't. So
Right.
although you have a rhizobia population in the soil, some will possess a genetic element. In the case of peas and beans, we call it the symplasmid, symbiotic plasmid. And that will allow it to recognize the legume. There'll be a host rhizobia recognition that goes on. The traditional method of infection is that the root hairs will curl. It will trap the rhizobia in the hook, if you like, of the root
Right.
hair. The rhizobia will infect down the root hair into the cortex of the root.
Okay.
There it will stimulate cell division. And you'll have quite a sophisticated structure that forms with a zone around the outside, which is not infected, with the rhizobia and a core in the middle, which is infected. If you were to cut the nodule in half, you'd see it had a red core. That red is like hemoglobin. It's a kind of type of plant blood that grabs the oxygen,
Okay.
and that oxygen is used to drive the activity of an enzyme called nitrogenase, and it's that nitrogenase that fixes nitrogen from the air into biologically useful nitrogen. Now, you're talking about my PhD area now, because that cortex, that uninfected band around the nodule core, to me is quite particularly clever, because the enzyme that fixes the nitrogen is damaged by atmospheric levels of oxygen.
Right.
So the cortex regulates oxygen diffusion into the nodule core so that the nodule core is kept at what we call microaerophilic levels, because if the oxygen level gets too high, the enzyme that fixes nitrogen is damaged.
Okay.
So the plant regulates oxygen diffusion to the core to keep the oxygen levels low so that nitrogenase can function, and it creates a diffusion barrier to vary. That barrier varies in its strength to let more or less oxygen through depending on demand. Very, very, very sophisticated system.
Yeah. So now you've done some recent work on that, I believe, haven't you? In terms of,
Well,
I mean, I guess what we're looking to do. From a farmer's point of view, if you can maximize that nodulation, well, you tell me, Pete. If are we looking to maximize that nodulation, or are we looking to optimize it? Perhaps might be a better term.
I think
And
we
how do we
are.
do that?
You have to be very careful what you wish for. So I'll answer your question in stages. Let's look towards one of the most successful legumes that we know, which is soybean.
Yeah.
Soybean is a success for many different reasons, but one of the main reasons is they've matched the elite rhizobia that nodulate soybean with the best genotypes.
Right.
Well, that's a bit easier to do in soybean, because in many places around the world, there are no rhizobia that naturally occur to nodulate soybean. Yeah, the center of diversity is in China. So there are places where soybean rhizobia exists, of course there is, but generally speaking, when it's been adapted for agriculture, you've had to apply the best inoculant to the best plant. So there's
Okay.
no rhizobial background, if you like, to compete with in the soil. So soybean is a very successful combination where you have the best plant matched with the best rhizobia going into an environment where the rhizobia does not already exist.
So when you say inoculate, so the seed has been inoculated with,
Yes,
right?
yes,
So
with a commercially supplied.
that's a little bit contrived, isn't it, Pete?
It's what's happened.
Yeah.
If you see, but on all our crops evolved. I mean, if you look at wild soybean, it's a sprawling plant. You would never recognise it as the
Right.
crop that we would see today, but people have adapted it, they've bred it, and it would be the same that's true for any of our crops, that they are rather contrived.
Mm.
What they are in the domesticated form is not how they appear now.
So, but
So
we don't inoculate our bean crops in the UK, for example, do we?
some businesses do, yeah, and there are some actually quite large businesses. It might not be fair for me to mention who they are, who are having success applying elite rhizobia to peas and beans in the UK.
Right. Well,
Um,
maybe
uh,
that's something we should be looking at in the Pulse Pioneer trials then.
yeah, we certainly worked with ADAS and the bean and pea yens before looking at rhizobia, a particular
Yeah.
technique to quantify the amount of rhizobia in the soil, and also to differentiate between those rhizobia that can nodulate and those which can't nodulate.
Right.
Um, And so that's been quite useful. There are certainly instances we find low yielding fields with low levels of nodulating rhizobia. So it might well be a solution for some. But also, if you look at the nature of the rhizobia themselves, we find that when we look in the laboratory or in a glasshouse study, that we can find 12 to 15-fold difference in plant growth depending on which rhizobia we put on the legume. Now,
Right.
it's one thing to see that in a lab, in a sterile environment, in a
Yeah.
controlled environment. Seeing it in the field is another issue, because then you're adding the rhizobia to the seed, and you're adding that inoculated seed then to an environment full of other rhizobia. And you want to be sure that the one that you put on the seed nodulates your crop, and that's another area of science where you're trying to maximize rhizobial survival on the seed and make sure.
Can I just establish here, Pete, that so this element of nodulation, is this crucial for the pulse crop? I mean, what I guess I'm getting to here is, can you get a more consistent yield if you have a better nodulation?
We have seen increases in yield, but I'll be honest with you, they've not been statistically significant. So while the yield average is higher across the number of replicates we've looked at, it's not statistically significant. However, I'll qualify that when I do an intercrop and I apply the leek rhizobia, I do get consistent yield increases.
Right.
So I suspect that when the non-legume is when the cereal is with the legume, that it's driving the legume just to fix that little bit better. And when you put the elite rhizobia on, you can. So in all my intercropping trials, while I've applied elite rhizobia, I see the yield increase. But let's take this back a step. I don't believe we've looked enough at whole crop forage values either, because what we can see, even in those plants that we see, we don't see a significant, what we call in science, a statistically significant increase in yield. We do see a higher average, but for our statistical purposes, in terms of peer-reviewed publications, we couldn't say that it was statistically different. But nevertheless, the yield is moving in the right direction, and the crops themselves are always more vigorous. So I feel there is a big biomass increase, but we haven't done enough research to, again, quantify statistically what that vigor improvement might be. And I feel there's actually more of an indication of yield increases using elite rhizobia for whole crop forage than for perhaps grain yield.
Right. I see. Well, maybe that, as I say, that's something that we can explore in some of our trials, and maybe this is something that some of our pulse pioneers would be interested in trialing in the field, and not just in the field, in the livestock diets as well, because you did just allude to it a minute ago about the differences that we're talking about that can happen with the value of pulses as a livestock feed. So do you want to just explore that a little bit more, Pete?
Well, I'm slightly concerned that we've been breeding some of these phytochemicals out of pulses, and I understand why that's been done. It's mainly been done with a view to serving pig and poultry markets. I'm not sure that breeding those things out is doing the best for ruminant gut health, potentially even also methane emissions from ruminants. So I'm curious about having pulses for different markets and maybe even different environmental functions. So at the moment, we're again using this One Health approach, and we're looking at some pulse types that are more phenolic and more tannic, and others that have had more of the polyphenols and tannins bred out. And we're looking at the impacts that has on human gut health, animal gut health, but also on mineralization rates in the soil, because we suspect that the old-fashioned, more lignified types probably have a longer in-soil half-life. So you'd have organic material in the soil for longer, where the mineralization rate was slower. Because you find these more woody types of legumes, for want of a better reason, while they might not be more digestible, that lack of digestibility or lack of immediate digestibility might be a good thing in other circumstances.
I see.
So it's as an ecologist, it's all about balance. And I don't feel you often get something for nothing. So if you drive things in a particular route, that's absolutely fine for that purpose. But it's nice to be aware of what you're trading off.
Well, I can I can see that we'd be very quickly led down a rabbit hole of plant breeding and so on, which is one of my big passions. But that's not really what we want to talk about today. And crikey, look at the time. We've already
But
been
it would
going
be
on.
nice, wouldn't it, to see legumes bred for different purposes and having specific
Yeah.
pulses for specific markets, I feel.
Oh, and with the
Yeah.
power of gene editing these days, you could identify the right gene and bring it in, and so on and so
Oh,
forth. But
now you've
let's
opened up another can,
Yeah,
haven't
exactly. Let's
you?
not go there for the moment. That's not the idea of this podcast, because in the very little time that we've got left, Pete, that I did want to talk about this element. You brought it up earlier about life cycle analysis, and I know that it's a very important part of the NCS project, and it's one that I think that James Hutton is leading on within the project. So, could you just briefly describe what you're looking to explore with this life cycle analysis, or what? Sorry, I ought to rephrase that. What elements that you're looking to bring into the
We're
project?
developing life cycle analysis for rotations. So it's not an analysis of the pulse crop per se, but it's about the utility of the pulse crop in the rotational setting. We hope it will be a useful addition to what the Farm Carbon Toolkit's doing within the project. They'll be very much focusing obviously on the farm carbon, but the life cycle analysis gives far more environmental impacts. Crucially, from my point of view, as I've touched upon earlier on, as well as having these impact metrics, we'll also have, for example, ecosystem function metrics or ecosystem benefit metrics. So
Right.
that might be lower pollution. So that's quite a new field of science, which is integrating system function metrics into the impact metric technique. And to me, that would be far more, as an ecologist, it's far more interesting to me to know how well the system functions, regarding of how good or bad you believe you're being to it. How's it actually functioning? So there's the nice, hopefully we can add value to the Farm Carbon Toolkit. We'll look at how our LCA technique fits with the Farm Carbon Toolkit in terms of our estimates. We also hope that the Farm Carbon Toolkit will inform what we do as well, and we'll add an extra dimension. So yeah, I mean, there's a lot of method development in there, a lot of data management in there. We're going to be gathering data not just from the CSC and from the pioneers, as he called them earlier. We're going to also have data coming in from a whole host of trials, because as well, we could then start looking at the rotational context and say, well, if a farmer then processes their pulses on farm, let's say for feed self-sufficiency, what might the added benefits be?
Yeah.
And we can go beyond the rotation to the whole farm benefits.
And so you're going to be looking at these whole farm benefits. Now, one of the terms that that I've heard about within the project that you referred to once or twice is this thing called crop goblin, which fascinates me, Pete. So do you want to just explain briefly what crop goblin?
Yeah, in simple terms, it's a scenario generator. And what we're going to do is develop a scenario generator for crop rotations. So if you can imagine, you can give this model a directive, which is say, I want to be climate neutral by 2050.
Right.
Given with the new information we've gathered within the project, and we look at different regions of the UK, which will have different land use capabilities, different rotations fit in different parts of the UK. We could ask the crop goblin, as we're going to be calling it, to generate scenarios just randomly, different crop rotation scenarios relative to bioregions or crop land use capability maps. And then we'll say to crop goblin, but which of those will give us the lowest carbon footprints?
I see.
And it will tell us what rotations will be best where and for how long. So in that sense, it's a design tool to perhaps inform governments where it might target the balance and nature of post-cropping.
Yeah, and perhaps provide incentives for going down a particular route, but at the same time for farmers to see how they can best capture carbon within their rotation, perhaps.
Yeah, it's already been used. Goblin itself, the first version, has been used in Ireland. It's received quite a lot of funding from the Environmental Protection Agency. And they've been looking, obviously Ireland has a particular challenge with greenhouse gases because it's 35% actually of the national emissions as opposed to 25% in many other developed countries. That's because they're big meat and dairy sectors. So
Right. Yes, of
so,
course.
so they have to look at how they're managing their land
Yeah.
particularly carefully, and that can generate scenarios with not with regard to just what you might crop, but also with regards to how you balance the production of pulses with other land uses such as
Mm.
wetlands, agroforestry,
Right.
and things like that. So you can then put your pulse production into the context of rewilding, carbon sequestration,
Yep.
and other things. So but anyway, we're not going down the full goblin route. We're going down crop goblin, which is to do with pulse-based crop rotation. So that will be the focus here. It will be a subcomponent, if you like, of the main goblin model.
Fantastic. Well, really looking forward to that coming up. Before we move off this subject of, because you mentioned data management and the enormous amount of data that we're bringing together on this project, and this is a hot topic of conversation with farmers in terms of, who owns the data, who controls the data, what's my data being used for. So, do you want to just, talk me through some of the data that we're bringing together on this project, and how we're using it, and using it for the benefit of the farmers who are contributing their data rather than against them.
As I understand things, Tom, the farmers remain the owner of the data, and so we only do what they give us permission for. So there will be agreements that will be signed and agreed. I mean, from my point of view as a scientist, particularly when public money is involved, I think it's fair and right that the public money is being used to generate data. Then it's right that that data is made available to the public who have funded it. But also you have to bring into question what value is a small data set, on its own. If you can have people working together in a collaborative fashion, you can really build some really unparalleled and quite extensive data sets that really reveal the complexity of the world that we live in, and how best we might manage it. So to be part of a pioneering group that provides a unique data set, I don't think there's been a rotational trial that I'm aware of in the UK of this scale,
No.
where you can take pulses and look at obviously the pulse cropping itself, then look at the effect of the follow-on crop, and then have it analysed using the Farm Carbon Toolkit and perhaps even a life cycle analysis tool as well. Individually, farmers will get to know the systems better. We'll have a better picture of what pulses will do in terms of yield uplift to the following crops and an input offset. So, who wouldn't want to be part of a group that delivers something as wonderful as that?
Yeah, absolutely. Well, that leads me nicely into. So, if anyone would like to get involved, if you go to ncsproject.co.uk, that's ncsproject.co.uk. There's more details on there about how to join the Pulse PEP, which is our online platform where people are bringing their experiences. And we've got the Pulse Pioneers now who are just signing up, who will be bringing their trials to the Pulse PEP. And leading the discussion on how to get these yield consistency benefits that Pete and I have been talking about, and bringing some of the benefits from the various scientific platforms that we're looking at. Just to reassure everyone, in terms of on the data management side. So we've looked in close detail at this, Pete, haven't we? We've had many conversations and meetings about this. How are we going to treat farmers' data? We've distilled it down into a set of data principles, and all of our farmers, the Pulse Pioneers, will be told what the data principles are, what we will do with their data, what we won't do with their data, and we have drawn it up in the best sort of possible light to make sure that it benefits the farmers who are involved, but does not compromise their data sovereignty. But listen, I think that's just about what we've got time for, unfortunately, Pete. As I say, anyone listening who wants to get involved in the project, do please go to ncsproject.co.uk, sign up to the Pulse PEP, and that's where it's all happening. Pete has got lots of contributions that he's been making to the Pulse PEP, and it will continue to make lots of contributions. And if anyone has got any specific questions of Pete, please go to the Pulse PEP, put them on there, and if Pete doesn't see them, then we'll prod him, and we'll get him to answer them. So we are looking after that forum. Sky, my colleague, is moderating it, making sure that if people post any questions, that they are answered by all the scientists. And we've got many of them involved on this project as well. So listen, Pete, thank you so much for joining us today. That was a really fascinating deep dive into pulses. And in particular, I enjoyed the conversation about rhizobia and nodulation. That's a real fascination of mine. And I know that there are many listeners who share that fascination as well. Not that I want to sound like we're a bunch of saddos. But anyway.
Thanks very much to you too, Tom. Thanks for the chat. I appreciate that.
No, not at all. Well, before I go, though, I just want to ask, so what's your key ambition from this, both for yourself, but also for UK farming? What can we achieve here, do you think, Pete?
From my point of view, I'd like to see what I refer to as ecological intensification, where we're protecting the system that's nurturing our food production. And I think for farmers, what I hope for them is that I would like to give them more agency. Whether it's the tools to better manage the system, I would like to increase their input dependencies so that we can achieve more ecological food systems.
Yeah. No, that's absolutely chimes with what we're looking to do with Boffin as well. We have what we say is knowledge has value, and I think farmers don't appreciate just how valuable their knowledge is, but it's only valuable if you share it, and that's what we're all about in this. And the great thing about this particular project, Pete, is that we've got wonderful scientists like yourself involved as well. And so the more we can all share that knowledge, then we can build that knowledge and bring it on. And especially
you're spot on, Tom. It's all about the discussion. Yeah. I read papers, and it's nice to read papers, but actually scientists talking to scientists and farmers talking to scientists, farmers talking to farmers, it's all about the narrative. So I completely agree with you there, Tom.
And it's all happening on Pulse Pet. So join the conversation. Please do. It doesn't end when this podcast ends. It only starts. Right. Great. Well, thank you very much, Pete. Thank you very much for joining us. And join us again on the Boffin Buzz with our next episode. Thank you and goodbye.